is Andres Horta, and today we'll be talking about the mitochondrial electron transport chain. So the electron transport chain occurs in the mitochondria, specifically in the inner membrane. This is the final stage of cellular respiration. So to begin, 3 of the electron-carrying molecules in the ETC are ubiquinone, iron-sulfur proteins, and cytochrome C. Ubiquinone, which is also labeled as CoQ in this diagram, or it can also be labeled as Q, is a hydrophobic molecule, meaning that it can travel through this hydrophobic region of the inner membrane. So in its fully reduced form, ubiquinone becomes ubiquinol, which it could be labeled as QH2. During the electron transport chain, electrons move from NADH through the 4 complexes to create a proton gradient that drives ATP synthesis. So as you can see here, the inner membrane space, also known as the P side, is high in protons, while the mitochondrial matrix, also known as the N side, is high in electrons. This gradient drives ATP synthesis for the formation of ATP. As a reminder, NADH and FADH2 come from different complexes from the mitochondrial matrix, such as the citric acid cycle and the pyruvate dehydrogenase complex. So complex 1 is also known as NADH dehydrogenase, and this is because the enzyme will be oxidizing this NADH molecule and turning it into NAD+. So complex 1 begins, like I said, by catalyzing the transfer of the hydrogen ions from NADH onto flavin mononucleotide, which is labeled here as FMN. So NADH is oxidized into NAD+, and FMN is reduced. This NAD+ molecule can be reused by the citric acid cycle or by glycolysis. And as a reminder, this NADH molecule comes from cycles within the matrix, like the citric acid cycle, that generate these molecules. So the electrons move from this FMN molecule along these series of iron cluster centers. This creates an electron current which powers the pumping of these hydrogen ions. Complex 1 is a proton pump. In this step, the complex pumps 4 protons across the inner membrane space, as it can be seen right here. So this flow of electrons powers the pump of these 4 protons. Ultimately, this begins to establish the proton gradient that will power ATP synthesis later on. So as electrons are flowing through these series of iron sulfur centers, they will ultimately bind to ubiquinone. And in combination with these 2 hydrogen ions, it will be reduced into ubiquinol. Complex 2 is also known as succinate reductase, and within this complex we can find succinate dehydrogenase, which had been previously seen in the citric acid cycle. So if we recall, in the citric acid cycle, succinate dehydrogenase will actually oxidize succinate into fumarate, forming FAD2H2, the reduced form of FAD. So as we can see here, this is the binding site for succinate. It will be oxidized into fumarate, and this FAD will be reduced into FADH2. And now we again see the flow of electrons through these this iron sulfur centers, and will ultimately reduce ubiquinone into ubiquinol 1. So now that both complexes have formed these ubiquinol molecules, they will detach from the complexes and move on to the next step, which is complex 3. So now that we have the reduced ubiquinone, which is ubiquinol, from complex 1 and 2, we will find that it is going on to attach to complex 3, which is right here. And complex 3 is also known as cytochrome reductase. This is because the main goal of complex 3 is to reduce cytochrome C from the ubiquinol electrons that were brought in. So again, the purpose is to transfer the electrons from ubiquinol from complex 1 and 2 to the cytochrome C. And in this complex, we'll find that there are 3 subunits. There's cytochrome C1, which is right here. And cytochrome C1 contains two heme groups. Sorry, it contains one heme group. And there's also cytochrome B, and this one contains 2 heme groups. So lastly, there's also these 2 iron 2 sulfur centers from the RISC iron sulfur proteins, which are located right here. So the QN and QP are the 2 binding sites for ubiquinol. So this complex undergoes ubiquinol to go through the Q cycle. So there are 2 processes to this Q cycle, and one of them is that ubiquinol will bind to one of the bind sites, and it will be oxidized by this iron Fe3 plus, and it'll be that will be reduced to Fe2 plus. Now, the electron will flow onto the cytochrome C1 heme, and then that will bind to the cytochrome C, reducing it into the reduced form of cytochrome C, which will detach from the complex 3, and we'll move on to complex 4. This is only one of the electrons from ubiquinol, and what happens to the other electron is that it will reduce the cytochrome B, and this cytochrome B will reduce one of the ubiquinone molecules into a semiquinone, which is the reduced form of ubiquinone that with only 1 electron rather than 2. So now that we have established that 1 ubiquinol will come and be essentially will reduce this cytochrome C to release it, in the second step of the Q cycle, we see that another ubiquinol will come in and undergo the exact same process, ultimately reducing another cytochrome C and also reducing the ubiquinol that hadn't been fully reduced and making it fully reduced to ubiquinol rather than the semiquinol. And in totality, what we find is that the 2 ubiquinols that were oxidized into ubiquinone will release their 2 hydrogen ions, and each time releasing or pumping 2 hydrogen ions, 2 protons across into the inner membrane. So since this occurs twice, we get a net of 4 total protons being pumped into the inner membrane space. Now that we have these reduced cytochrome C molecules, they will go on to the next complex 4. Complex 4 is known as cytochrome C oxidase, and the goal of this complex is to transfer the electrons from the reduced cytochrome C molecules to oxygen molecules. So this is one of the main reasons that cells need oxygen to complete this cycle. So the complex is composed of heme A and heme A3, as well as 3 copper atoms. Cytochrome C will bind to the complex, and then it will transfer an electron onto copper A and the copper A center. The electron is then transferred to a heme A, and then from heme A, it is transferred onto heme A3. Heme A3 then reduces copper B. So now that we have a reduced form of copper B, another cytochrome C will bind and reduce again, the copper AA center. Then that will reduce the heme A, and that will reduce the heme A3. Now we have the heme A3 reduced as well as the copper B reduced. So now every time the cytochrome C is oxidized, it unbinds and releases. So now that we have the reduced forms, oxygen will then bind to the heme A3 and the copper B, and it will form a peroxide bridge between them. And then 2 more cytochrome C molecules will bind and reduce back to the copper B and the heme A, and 2 protons from the matrix will be introduced, and they will attach to the heme A3 and copper B. This will break the peroxide bridge. Now this forms a copper B hydroxide as well as a heme A3 hydroxide. So now that we have these 2 hydroxides, 2 more protons, 2 more hydrogens are introduced from the matrix, and they will bind to the hydroxides, and they will create 2 water molecules. So now the movement of these electrons through the complex 4 will cause a total of 4 protons to be pumped across the 4th complex. And to conclude, we will have a total of 10 protons pumped across all the complexes, forming the proton gradient across the inner membrane. Thanks for watching.